Welcome back to Biotech Blueprint. Today's episode is part two of our special series on gene therapies, co-produced with the Biotech Capital Compass and Hartaj Singh, calling in from Brooklyn, New York. If you missed the first episode, no worries. You can find it on our Substacks, on Spotify, on YouTube. This one focused on CRISPR and gene editing. But if you're more interested in gene therapies beyond CRISPR, you're in the right place today. As a summary, in the first episode published last week on our Substacks, we focused on gene editing, like I said, especially CRISPR, how it's reshaping science, medicine, and the biotech market. We explored CRISPR's rapid rise, its advantages over some of the earlier tools that we might talk about today, and its transition from lab bench to clinic, culminating in the FDA approval of Casgevy for sickle cell disease. We covered the differences between gene editing and broader gene therapies. We also talked about landmark partnerships and investments, emerging regulatory frameworks, and key challenges still ahead, such as off-target effects, deliveries, durability, and scale-up. Today, we want to focus on the science behind non-editing gene therapies, the investor perspective on where the field is heading, and of course, we'll talk about Sarepta, whose roller coaster over the past 2 weeks really shows both the promise and the complexity of this space. But before we dig in, Hartaj, perhaps you can start by giving us your broad opinion on gene therapies and talk a little bit about your experience before I do the same and talk a little bit more about the science maybe behind gene therapies.
Yeah, Katerina, thank you so much for the welcome and the really nice synopsis of our last call. It was really a lot of fun.
Mm-hmm.
I'm trying to lean back a little bit for this call because it seems like at the previous call I was right in people's faces. So, I'm gonna sit back a little and try to give people a little bit of room who are, a little space who are listening to this in New York. If you have problems with space, meaning you don't like people butting in on your space, New York is probably not the place for you. So that's why I'm probably happy, doing a call like this. But for everybody else outside New York, we want to keep it easy going. But no, thank you. Look, I think from a Wall Street perspective, and I work in clinical drug development also, and I think you'll probably touch on this, and you touched on this last time. Gene therapy seems to be the broader category, and then gene editing the smaller within it. But for purposes of simplicity, especially on Wall Street, and I think even probably in the corporate setting for telling that story, these are very complicated stories. Gene editing, gene therapy are very complicated. Generally speaking, in Wall Street, we tend to bucket companies, and gene therapy is usually regarded as companies that are using some kind of like a vector or some sort of a delivery mechanism to get, whether it's a snippet of a gene of interest or something else, or Cas9, or the enzyme that can cut things, or other aspects into a cell, whether nucleus, cytoplasm. And then gene editing is specifically for those companies that are going right into the nucleus and editing those cells. Of course, mRNA is different. So I think in that context, we tend to simplify it, which is why when somebody says gene therapy to me versus gene editing, I view them separately, even though you can say that there's a lot of overlap. Yep. And, I think it would be really helpful, to us before we kind of talk about the clinical or even the preclinical, because I think both these areas, gene editing less so, but gene therapy has had a long history. I mean, as long as, physicians and, scientists have been observing the human condition and looking at viruses and how viruses propagate, there's been this idea, right, that you could use an inactive virus or something like it as a delivery mechanism, but it had a tremendous development time, right? The science has had a long time translating to the clinic, right, Katerina, because of certain issues.
Yeah, so I can talk a little bit about the science now, maybe, and kind of you can always jump in and we can discuss what I talk about, but. You're right. It took a long time, and in the non-editing gene therapy, there's a functional gene, right, that is added to or introduced without directly cutting into the DNA, like CRISPR. And we talked about this a little bit last time, but the clinical trials, I think, with any sort of gene therapy began in the 1990s. And we talk a little bit about Jesse Gelsinger and so I'm not going to talk about it again. But these setbacks, like Jesse Gelsinger and then later cases, which were actually something we can discuss later with the sort of limitations of gene therapies, were vector-related leukemias that also paused the field for a very long time. And really, the newer clinical trials and sort of the revived gene therapy begun in the 2010s, really. So it's not that old of a technology when you think about it that way. But like you said, the vectors that are used, that's much older of a technology, but repurposing it for gene therapies was really done in the 2010s.
And
And so,
Katerina, just to give you like a real that's a great insight, because I will tell you what, we'll talk about Sarepta more later, but just to give you an idea, their vector that they've used for the delivery of their therapies for DMD, which are just shortened forms of that of that gene that has that vector, what's called AAV R874, was developed for a long time. I believe it's a gorilla adenovector, and it was discovered at Children's. This is I think the hospital associated with Ohio State University. I could be wrong here. My memory might be getting a little stale, but they spent 10, 15 years, developing that vector and doing a lot of work around, the risks, the safety issues there. For that, because there were a lot of ongoing issues in DMD with vectors like AAV9, which were being used by some companies, et cetera. But to your point, going from identifying a vector that can be used as this payload, right? It's like the outside and it's empty shell inside. You put in whatever is your, right, takes a long time.
yeah, and also something we can touch on. So I'm an immunologist. That's what I did in my PhD. But the reason why we can't really use adenoviruses and why there's some issues with AAV also is the pre-existing immunity. So you basically have pre-existing immunity because as children especially as children, infected by adenoviruses. There are, a lot of colds are adenoviruses, and so we have a strong pre-existing immunity against these vectors. Even AAV, even though that's not really a disease-causing virus, it kind of co-infects people. And so you don't really want to have or use like human adenoviruses because the thing is that we will mount a strong immune response against it with our antibodies, form these complexes that can trigger a very strong immune response. So that's like one disadvantage that I'm sure, and I don't know the history very much, but I'm sure they battled with, and so that's probably the reason why they use the gorilla AAV.
No, you're absolutely right. You're absolutely right, Katerina. In fact, even with the gorilla, so just to give you an idea, the AAV9, for example, about 1 in 4 patients has like already antibodies against it in their bloodstream for whatever reason. R874, Sarepta's vector, because of its uniqueness and had not been really used before. They actually were showing autoantibodies only in about 14% of patients, and that stayed pretty steady. And that's actually pretty good, for 1 in 7, compared to, as you said, other vectors that, where there's a big immune response because you might already, have antibodies to.
Yeah, so the reason for that is not that you were infected by a gorilla virus, it's because of cross-reactivity. So basically, some of these, monkeys are similar to humans, and so some of these viruses basically are very similar. And so the antibodies that are created are kind of not as specific, right? And so then they can kind of cross-react to other viruses that look similar to what you have encountered. And so that's the reason. I was part of this vaccine trial that we did at Hopkins, and we used like a chimpanzee one. And I think that the response to that, the trial ultimately failed, but I think about 28% of people had response, like had antibodies against the vector, which is obviously not, ideal when you're trying to vaccinate them. So that trial ultimately failed, but it's been shown in literature that around the 28% is okay for a vaccine to actually work. But that's just a side note. As we discussed last time, right, we use the viral particles because they're really good at getting the cell inside of the cell. And so most approved gene therapies use these viral vectors. And like we said, there's 3, but the adenoviruses, they're not really used because they just mount such strong immune responses that honestly there is just not Many clinical trials use them after the first sort of setbacks. So the two that we have, as you mentioned, is AAV and then also retroviruses and lentiviruses, which we can touch on a little bit, but they have their own setbacks, their own advantages, and some companies have used them, and there's actually approved FDA therapies that use lentiviruses. So yeah, AAV has been hugely popular in vivo gene therapies because they tend to usually cause only mild initial immune response compared to the adenoviruses. They don't insert in the genomes at high rates like lentiviruses do, so there's a sort of reduced risk of insertional mutagenesis, which we can discuss a little bit later. But the example, like you said, is Sarepta's Elevidys, Novartis's Zolgensma for SMA, and others. There's Unicur's drug, I forget the name, but it's I think for hemophilia B, and so there's a bunch of these approved gene therapies for that use this AAV technology. And like I said, the advantages, I mean, there's a lot of advantages. It can provide sustained gene expression with a single dose. You know, certain serotypes, like you said, like serotypes meaning, you know, these they originate in different animals or different sources, and they can actually target specific organs, which is useful, right? And like I said, they usually should cause mild inflammation, making it relatively safe in many contexts. And of course, the flip side is that they're still foreign to the body, these vectors. And so especially when they're given in such large doses, like we mentioned last time, with Elevidys, for example, it can really, and these doses are really systemic. I mean, it can kind of be a major issue. And as I said, one of these issues are the pre-existing antibodies, the complexes that can form and then really induce a very strong immune response. So they're very immunogenic. They can activate the innate immune system, but also the adaptive immune system, which I think, or we think, is the issue around the liver toxicity. So they often mount a strong CD8 or cytotoxic T-cell response. And, you know, in the modern trials that we've seen, patients are usually immunosuppressed so that this doesn't happen. Usually they're on corticosteroids, or Sarepta, I think, wanted to put patients on sirolimus, which is a T-cell inhibitor. But again, the most severe AAV toxicity, which is sort of universal for these AAV therapies, is the liver toxicity, which we think is because of CD8 T-cell overactivation. And then that can, you know, cause a lot of inflammation. These cells accumulate in the liver, they cause a lot of inflammation, and then that can lead to liver failure, which was the case for those two boys who passed away in the Elevidys trial.
Yeah, and Katerina, it's interesting, you know, again, just using Sarepta as an example in this case, in a little bit of context, right? So this was a company that has you know, these antisense oligonucleotides originally that were approved in 2016. They were actually very controversial. If you go back, it was probably as controversial as what's going on to today. This company has had more than its probably share of controversies, but they're dedicated, you know, to DMD. And that was when I started covering this company, actually, and I, you know, the drug was already approved, their original antisense oligonucleotide. And the stock was trading around 10 or 15 bucks. And I started Oppenheimer, and I said, you know, I actually think this drug could be beneficial for patients if you look at, you know, longer term data. I did work in clinical trials in small numbers. And we actually ended up being right. The stock went up to about like 70, 75, 80 bucks. And this was around the time in 2017, 18, when Elevidys, I forget what its scientific name was, but came, you know, under development. And to your point, it's actually interesting because I went to a neutral on the company at that point because I thought the benefits early on, you know, were pretty clear from, you know, in vitro data they were showing like dystrophin expression, what's called microdystrophin, et cetera. But based on the history, you know, many decades around gene therapies and delivery problems with these delivery mechanisms, you know, we felt that, hey, you know, we need to wait longer and see more data. We waited 4 years before, you know, we became, you know, sort of like, bullish on the company again. I was probably a little bit of persona non grata, you know, among some members of like the biotech investment community, but Sarepta at that time was a consensus buy, you know, and but we did a lot of work. And actually, by the way, when on the safety side of this R874 AAV vector, when we upgraded it, you know, I believe it was in 23, we did it with the belief that the safety profile for the ambulant boys was actually pretty good. was that they had shown efficacy over many years, and then they had boys that have been on this drug for 5, 6 years, and they had come up with this dosing regimen. They don't use sirolimus now, but for the ambulant boys, they use prednisone. So they give prednisone when they give this therapy because it homes into the liver, right? And this AAV vector. And so they give prednisone because there's 30 to 40% of the boys show elevated liver enzymes. They give the steroid prednisone for a certain number of weeks, and it puts that down. So the ambulant boys who've been treated on this drug for many years really haven't shown any issues. There've been some hospitalizations, but to your point, it's the non-ambulant boys that seem to be, or in this case, young men, the two that passed away, and there they didn't run a trial. We can talk about that later, but they didn't run a trial there. They got what's called accelerated approval, and that's where the problems are arising now. But to your point about that immunosuppressive regimen, in this case, Sarepta took 5 years figuring that out, which is why the therapy got approved for the ambulant boys, as I believe it should have and still should be.
Yeah, that's a great point. There's going to be some sort of elevation in liver enzymes, and so putting them on this corticosteroid for a bit, for some amount of time, is basically going to suppress T cells while the vector does its work, and then they can kind of wean them off, and it's very transient, right? So that's the norm. The usual response is fairly mild liver inflammation. But like you said, the patients, the non-ambulatory patients who are kind of the worst cases, right? I mean, they're probably their bodies are probably not in the best shape to begin with, and so I wonder if that contributes also. And I'm sure they were also put on these corticosteroids, but maybe it's not sufficient. Maybe their bodies are just in a state that cannot support, in some cases, this therapy.
No, Katerina, it's actually amazing that you say that, because you were talking about the T cell issues, right, in the liver and stuff. Because I actually did some research, this morning, and we had done it, previously when we covered Sarepta, on if there was a clear correlation between, as a boy's age, their liver functions get worse. There's actually almost no study out there. There's some kind of circumstantial evidence that suggests that livers over time, because as the muscle is breaking down, it has to be metabolized, right? And it's metabolized and then excreted, I think either through the liver or through the kidneys. And so that puts a big burden on the liver. The problem is that most of the DMD boys, when they die in their 20s or 30s, they usually die because of cardiomyopathy. So that's heart is a muscle, and it fails, right? So you don't have the time to see if other organ systems are being affected. I don't know if pathology is ever done on other organs for these boys, but the published data is very little. But to your point, I think it's fascinating you're talking about T cells, because they probably want to use sirolimus in this new trial that they suggested with the non-ambulant, is because they need a more powerful immunosuppressive regimen, because apparently with these non-ambulant boys, prednisone was not doing a good enough job to keep the elevated LFTs in the liver of these boys, which probably their livers were in worse shape because they're already, they're bedridden. They've had the disease their entire lives. So their organs are in worse shape. So I think there's probably some circumstantial, evidence to that point, but no direct correlation, I would say.
Yeah. Yeah, and as you mentioned, sirolimus is it's an mTOR inhibitor, so it directly inhibits T cells, and it's pretty strong stuff. It's given to patients that are after organ transplant surgery. So it's pretty strong to make sure that their immune systems don't reject the transplanted organ. So it's like a strong immunosuppressant. But yeah, I also note. So you mentioned the dystrophin, the microdystrophin levels that were successfully making it to the muscle cells, right, in this case. So I was doing a little bit of research on this again, and as developing, and it was kind of news coverage. So I'm not sure if they're experts on this, but they mentioned something about, and they were feeling very strongly about this, is that the clinical benefit of Elevidys has always been in question, in a sense that the FDA, I think they said that the microdystrophin expression in the muscle cells did not correlate with an improvement necessarily of symptoms. And so even though later on then there was clear clinical benefit, it was kind of murky, some of it. Some of the data was not statistically significant. What was your opinion on that?
Katerina, with the advisory committee meeting for Elevidys, the randomized, blinded trial didn't show really a benefit. And that is what the committee looked at. Its mandate was very clear. The committee members, they had to vote specifically on the randomized, double-blinded like data of which the trial ran for a year. Now, what's interesting is that the committee did have a mandate to look at data that the company had generated where these boys that had been on this drug and on clinical trials for many years. That wasn't randomized. They went into long-term extension. But over there, the drug clearly showed a benefit. And the way you could see it was like, there's a well-known natural history progression of boys on DMD. And if you took all the boys who are on Elevidys, they just were not losing as much muscle function over, year 2, 3, 4, 5. Whereas natural history, where these studies have been done, many University of California, I believe it up in Northern California, I'm forgetting, they have a center there where they've done a lot of work on natural history progression. And, Dr. McDonald, I believe his name is, and, they have a lot of data that shows natural history progression of boys going down over time. But like boys on Elevidys over year 2, 3, 4, we're still losing function, but at a much slower rate. And the advisory committee, members never looked at that, right? So we just kind of think of it this way. Think of it, it's a little unfair. And I don't want to prejudice my answer here, but let's just say for sake of argument that I'm taking the SAT, right? And I'm dyslexic, right? No matter how brilliant I could be, I'm going to do bad, right? And if the committee does not take into account my dyslexia, then I will just look like I'm a bad student or I don't know how to take exams well, right? Standardized exams. So, I think in some ways the dice was loaded against Sarepta at that advisory committee meeting. And it's easy to say that the drug didn't work because yes, in year 1, it showed maybe a clinic didn't show a statistically significant benefit, but over year 2, 3, 4, the benefit got better and better and better. And they just didn't have a few boys worth of data. They had over 100 boys worth of data at that point. So which is why I always believe that, these therapies, because this disease is so debilitating and progresses, right? Just to actually kind of start not even halting the disease, but slowing it down is such a hard task, right? That it takes a while for a drug to show that. And then the FDA giving them only a year against blinded, patients to show this, that's again, in rare diseases, that's a very high ask. And if the FDA had sold a company, hey, you got to run a 3-year trial, like there's no way investors wouldn't have given them enough money for that, So it's just kind of like this, like they're stuck between a rock and a hard place. Companies that are developing drugs in these rare disease spaces, there's only 10,000 boys or plus or minus 5,000, with DMD in the United States. So like, how do you develop that, right? And so I think if you look at the whole picture, and I believe right now the FDA will, because there won't be advisory committee meeting that's going on, but they look at all the data in ambulatory boys, and how well and how many years of prednisone usage, immunosuppressant has happened, and they'll show them, Sarepta will be able to present them all the follow-up data on boys on long-term Elevidys. I think most likely FDA will come back and say, what, probably for at least the ambulant boys, this drug should stay on the market. At least I think if they look at the data and they look at it objectively, or the entire data, not just a blinded clinical trial for 1 year, I think most people will say this drug, has an effect and it gets better over time.
Yeah, no, I agree. And before we get into the FDA response a little bit, I wanted to ask you. So I know that Sarepta or Elevidys missed its, there were kind of these misses and hits and misses, right? But I know it, for example, missed its primary endpoint in phase 3, even though the biomarker data looks solid. Do you think that because there's the benefit is clearly there, do you think that this kind of reflects like a clinical development plan issue for the company, maybe choosing the wrong endpoint or underestimating variability, or is it just, part of a risk of working with rare diseases, in your opinion?
I think your last point, I think, is most relevant. It's a risk of working in rare diseases. I mean, let's just go back, right? Let's just go back and use an example. There's a company that I admire greatly, and they have done a tremendous benefit in the cystic fibrosis space called Vertex, right? And they have these oral, molecules that help the cystic fibrosis protein misshapes itself, and they kind of prop it up, And the first, for a tiny, subset of the population, the drug was, gave about a 10% benefit, in terms of like improvement. And you would say, oh, wow, 10%. If you had a migraine, for example, right? Let's just say right now you're having a migraine, and you know how bad a migraine can be if you've had them. I've had them from time to time. Or you're just having a really bad headache. And I give you a pill, and I say, oh, it's going to get 10% better. You'll be like, thank you, but no thank you. I don't need this pill if there's only going to decrease my pain by 10%, right? But to the person who's bedridden or a teenager with cystic fibrosis, who can't go to high school, can't participate, like 10% is a huge thing. It's the difference between getting out of bed and being able to go to high school and not getting out of bed. It's the same thing here. This Elevidys and their antisense oligonucleotides provide a small benefit, absolutely. But these patients are in absolutely the worst condition, and their diseases are rapidly progressing, right? Now, to your point, I think that there's a lot of people, both companies and investors, that probably were not happy with Sarepta and still are not, because many people felt that Sarepta got out of, and I hate to use this term, but it's like Monopoly. They got a get out of jail free card by the FDA, which is like, okay, you can get approved based on this trial that didn't work, and then FDA says no to everybody else, right? Where trials don't work. So I have met clients, when I worked on Wall Street, I met clients and companies that were not happy with that because they felt like the rules were getting bended for Sarepta. But if you then had the conversation that the rules weren't getting bended for Sarepta, they were getting bended for the DMD patient population. This disease is massively progressive. By the time these boys are 12, 13, they're bedridden. Their minds are active, but their entire body is deteriorating. Think of that. It is the worst prison one can envision in life. Your mind is fully active, but your body is deteriorating around you and you can't do anything about it, right? Like you're in a prison, a complete, prison. And so it's not the company that got this, get out of jail free card. It's the patients and their caregivers and their families. And I think if we layer in that way of thinking, then maybe, what the FDA did for Sarepta was, I would say, not that bad a thing. Now, of course, 3 people have died on this drug, but they weren't, the 2 people who died were not for the, almost a decade worth of development FDA did for the ambulant boys. There, nobody has died, right? And they seem to be doing fine. Most of the boys there are some issues from time to time, as with any drug, like we talked about aspirin. You take it, there's a 1 in a million chance you get a heart attack, right? That's what the label says. And the other man who died at 51 years old was for a therapy that was in development still, right? So they're still figuring it out. So I think the issue is that the FDA hurried up and the company requested it for approval for the non-ambulant boys when maybe they should have waited a year, 2 years and done a study, even a small study, and to figure out that these boys that are much more fragile, is it worth it giving them this huge amount of AAV vector with prednisone, or should we use like other approaches? But again, hindsight is always 2020.
Right. Yeah, that's a great point. Like you make a great point there that they kind of have to follow certain rules, right, as well. Like they can just bend rules for this and that, and like I see the point. What do you think about the FDA response? So let's just maybe summarize what happened. So the FDA was, even since the beginning, divided. There was a tie, right, when they were voting for Elevidys. But now, after the 2 deaths, and then a sort of unrelated death in the limb-girdle muscular dystrophy, which still uses the same delivery system, this was kind of like the last straw. There were some opinions that, and we sort of talked about this before recording the podcast, that Sarepta reported it late, which you can maybe give me your opinion on that again. And I know your opinion, but I would like others to hear it. They reported this like a month later after this 3rd death occurred. And so the FDA maybe was angered by that a little bit and said, like, let's pause shipments for Elevidys. And the company first said, no, we haven't seen any adverse effects for ambulatory patients, right? Like you said, they're all mostly benefiting. And so there's no reason to pause shipments. But then eventually they had to comply. I'm assuming that the FDA put pressure on the company in some way, and so that happened. Now the FDA didn't stop there, right? Because of the limb-girdle muscular dystrophy case, they also took away the platform recognition that they just had given them not long ago, saying that if they want to resume, they will need, and this is citing Endpoints News, but they said that there might be longer trials before they want to resume Elevidys shipments. So what do you think about that? I mean, it's a lot for Sarepta, and also the company just is restructuring as well, right? They're losing a lot of their workforce. Like, can they recover from this?
It's, and so absolutely, I mean, great question. Ultimately, from an investing perspective, this is the key question, right, Katarina? I mean, you gave a context for the key question is like, what's going to happen with this company going forward, especially in terms of like with all these issues. I mean, I was just looking at, what the analysts are concerned about. You've kind of like actually synopsize it in your own way, right? A lot of the analysts on Wall Street, I'm not a PhD or MD by training, but they're PhDs, MDs by training themselves. And, so they have your viewpoint, and you absolutely touched upon a lot of the stuff that, are out there in notes and reports about Sarepta. But let's take a step back. So there's two separate issues. The issue with the DMD boys, right? And the two deaths, which are in the non-ambulant population, right? And then there's a second issue, which LGMD, which is a different kind of a disease. There's two separate issues. There's one thing connecting these two issues, which is the AAV vector, R874. They're using it both for the ambulant and non-ambulant. It's the same drug, Elevidys, and then over here they used it as their delivery mechanism for whatever is the missing the genetic code for LGMD. It's called 2D. They were delivering that and that's what the 51-year-old man who had actually already had been dosed. Had gone through dosing everything and been past that now, and so died, later. And so on the first issue, look, FDA also revoked their what's called platform technology designation just recently. They only got it in June, and the designation was around AAV R874. So FDA said, okay, anything that's being developed with R874, we could probably give you look at it as like for potential accelerated approval because it's got all these years worth of safety and whatnot. Now FDA literally turns around 2 or 3 weeks later and says no. So that's rational because FDA is feeling like, all right, R874, because of the problems it's having with non-ambulant boys, and prednisone might not be enough. They might know in sirolimus, we've got to sort of take that back, So they kind of took away that platform therapy designation, literally, that they had given to Sarepta, which is rational. So it tells you that the agency's working rationally, right? Because that's a rational thing to do, right? Then in terms of pausing the shipments, I mean, the shipments are being paused both, for ambulant and non-ambulant boys, because most likely, there's been a lot of concerns among families. There's a Wall Street Journal article on this. There's articles, others coming out. Analysts are noting that, they're talking to people out there. So, and these boys, these are boys being treated, their families are their guardians, right? So their parents have lots of concerns around these boys, and they want their concerns to be assuaged. And how would your concerns be assuaged in this case? It's by a thorough review by a regulator of like the ambulant data first and foremost, right? And then in terms of non-ambulatory boys, if the parents and their boys want this therapy to be available, they probably will say, yeah, we want like at least a clinical trial for it to be run, and then FDA can decide whether they still want, access on a limited basis or not to the non-ambulant boys while that clinical trial is being run. And Sarepta is very good at running these clinical trials. They know the space inside out. So again, I think FDA is being rational there. They're taking a step back, saying, pause everything, let us take a look at the ambulant data, and we can come out and say, okay, if we let you resume the shipments of drug for ambulant boys, that's probably a thumbs up, to families, and saying, we've looked at all the data and we still think, yes, there's a risk benefit. There's a black box on the label now, but it looks for, at least for ambulant boys, this is acceptable, right? And then most likely non-ambulant, they'll tackle it separately. And then lastly, in terms of LGMD, look, that did not look well. They host a investor call, and then 2 days later, they go about and say that, oh, by the way, there's been a third death. They did not note that third death. Even they had known about it for a month. They said, this event had occurred, and they told the FDA. But look, probably, it was in clinical trial, so it was under informed consent. They had to get the regulatory approval. There was a bunch of things going on, and maybe on the day that they were talking about Elevidys, they just didn't have all these regulatory and legal, checkboxes signed off, right? And by the way, I remind a lot of people that the CEO is actually a lawyer by training, Doug Ingram. I believe, I don't want to misspeak, but he's a lawyer by training, and he's very sensitive to these issues. When I worked there, the company always was above and beyond. They always actually, even with me when I was a neutral on them and considered kind of like bearish on the name, they were always very transparent with me. They always had meetings with me. They always took calls with me. This is a very transparent company. And I think also having a CEO who's a lawyer by training and their corporate lawyers probably they felt that maybe we just didn't have the time to disclose that third death on the day of the call about the Elevidys deaths. And then 2 days later, they were able to declare it at that point because maybe certain checkboxes. I'm just purely speculating, and maybe I'm being too nice. I'm giving the company the benefit of the doubt. But I think based on their history with me and my experience with them, I would give them the benefit of the doubt that I think that they took these actions. Of course, it looks horrible. It does not screen well at all, and it looks like they have something to hide. But I think they've always seemed to be very transparent in my interactions with them. And I think this was just one of those, like, lemony snickets, a series of unfortunate incidents that led up to this. I mean, of course, it's horrible because people actually died. But again, that's my opinion based on just what has been disclosed, based on my history with the company and my own experience in drug development. I mean, I'm not a PhD or MD by training, but I worked 6-7 years in drug development. And so I think that there is logic behind what has happened here, and it's not some malfeasance on in terms of the company or in terms of the FDA. So that's my opinion.
No, I agree. It's just it doesn't look good. I agree with that. It's strange, right? But at the same time, and we talked about this a little bit, is why would they be hiding it?
Yeah, look, biotech and biopharma, pharma also has had a history of gotcha moments where drugs were shown, even became commercial, to shown to not have had some like issues or whatnot. Look, here's the thing, and this, right, as a scientist by training. If you give one aspirin, the aspirin label says that 1 in a million people can have a heart attack if you take it, right? So if you give it to one person, they might not have a heart attack. You give it to people, it might not, right? You give it to 1 million people, might see a heart attack, right? It's the law of big numbers, right? So that's what's happened in drug development, and is usually the case behind a lot of these gotcha moments. It's not that management is hiding something, it's that these drugs inherently have small signals that could be deleterious, and they don't show up in small populations. Before Elevidys was approved, it was given in like 100 to 300 patients, right? Now it's probably given to like 1,000 plus patients, right? So meaning, like, as you give a drug to more and more people, the potential for the harmful signal to show up just increases. I mean, that's basic statistics, right? How statistics works, right? So a lot of the gotcha moments, COVID vaccines, right? A lot of the gotcha moments, that have occurred in biopharma is because drugs were approved and then they were given to more and more patients, and then you saw like some kind of like death or something, And I'll tell you honestly, as an analyst, when I worked on Wall Street, when a drug got approved for the first 2-3 years, I always actually crossed both my fingers because I was like, oh man, it's going to be given to more and more patients. I hope something doesn't show up that was so tiny, didn't show up in clinical trials because of patient numbers, and now it's going to show up. There's a drug called Tysabri, for multiple sclerosis. Fantastic drug. Showed nothing. Look at, and then like for Biogen, it got approved, and then a year later, I was actually working, just started my career in Wall Street at that time at Lehman Brothers, and patients died, and it stayed on the market, but in a restricted format. Probably what will happen with Elevidys, to ambient patients, maybe not ambient, who knows, But I think these gotcha moments aren't because companies are technically, hiding something, quote unquote. I think it's because they're dealing with tremendous amounts of data, and as the drug is given to more and more patients, things, events show up that you could never see in small numbers of like patients originally. Now, I might be, simplifying complex issues, but
Yeah, and that's the issue with the rare diseases. Like, you have a limited patient population to do these clinical trials in, and then something can come up later as you're unfortunately giving it to a larger population. So yeah, thank you so much for your answers. It was very helpful and interesting. I always like to listen to your opinions and takes. Very, very cool. So that kind of concludes our episode today. We're on time, but next time we could cover the rest of these delivery systems like retroviral and lentiviral vectors. We could also talk a little bit about the new hot topic in the field, which is non-viral delivery systems like lipid nanoparticles and others. And we could also talk about other things. We always come up with new topics as we go and as we prepare. But yeah, any final thoughts? If not, we can kind of thank our listeners and yeah.
Sorry, Kevin. I want to make sure our listeners are in tip-top shape because that's why I flipped the screen. I want to make sure they were paying attention. Towards the end here, it's kind of like I feel like I have enough gray to feel like a teacher. I'm like, all right, let's just make sure that they're paying attention. I didn't want to do the in-your-face thing again, so I'm like, all right, I'm going to flip the screen and kind of wake them up a little bit. But no, it was always a pleasure, real pleasure, and thank you so much. I look forward to our next call. Thanks for listening to me. I think it's great. You always make me think, a lot and out of the box, and it's just a great collaboration that we have. And
I
I'm really
agree.
looking forward already to our next podcast.
Me too. I always have a great time and I'm excited. So yeah, thank you all for listening, for subscribing to Biotech Blueprint and Biotech Capital Compass. We appreciate your support and we'll see you next time.